Thank you, Peter. It is quite a pleasure to be here, and I thank you all, especially Dr. Raven, who invited me, but it's really quite a pleasure to be with such amazing thinkers. So I think that we are dealing with a lot of values when we talk about conservation. And I come from the agricultural community. I think I'll be forgiven for saying that I think in this particular room we might be talking about conservation as preaching to the choir. And in the agricultural community, I get a little bit more traction if I make appeals based on economics as a value, as opposed to some of the other values that we could be using to argue our case. Environment, a lot of producers consider themselves stewards of the environment as well. And certainly opportunity is a value that sells in my agricultural community. I think it's also important to remember that people are very resilient, and if we put this in the context of what's good for people, we'll get a little bit more traction. This is a saying from Woodrow Wilson, which was certainly before our Depression, but we had quite a bit of problems with poverty. And so poverty and conservation, I think, go hand in hand. So jump forward about 40 years from Woodrow Wilson, and agriculture had its green revolution. And that was the idea that if you could combine breeding with fertilizer, with good tillage practices, we could feed the world, and we could feed it really better than what we were. And I think what's really important, and I'll show in the next slide some of the data to support this, that before our green revolution, the idea was to put more land in agriculture to produce, and the philosophy now is to just intensify that agricultural use. So, Dr. Raven talked about 40% of the land mass being used for agriculture. It's actually going down. Here are some of the statistics. These are hot off the press from the World Bank. And because I am from North America, I use the North American data, but in my paper I gave you some others, some of the big cereal producers of the world. And yields are going up. This is a triumph for what we would call genetic resources. And I gotta say, I mean, we're talking about a natural resource. When I use genetic resources to our groups, they kind of get a headache. They don't like the word genetic, but we still try to use it. It's a little bit different than species for our community. But it's going up. Fertilizer companies would probably claim a lot of that yield, but we actually think it is about our genetic resources. And this graph is the graph of the land use. And you can see right around, well, you can't really see it because it's small, but right around the 1980s is when we started seeing a decrease in the land use, and that's an after effect of the Green Revolution, that we're actually taking land out of production. And this is happening in Europe. It's not happening everywhere, but the yield increases, I think, is going to sustainability of countries. And the United States right now is about, the World Bank statistics are out there, but that translates to about 35% of the land mass in the United States is now in agriculture, down from 42%. So I consider that a significant, a 7% decrease over the 30-year period is something that we can we can look forward to as a conservation mandate, that there is more land for other things to grow. Unfortunately, it's going to human develop, our American dream houses and turf. So when I try to describe genetic diversity and what diversity means in an agricultural context, this is one of my favorite slides. It's lettuce. People can get it at the grocery store, and they already know that there are different colors of lettuce leaves. Often they don't know about bolting and the seeding of lettuce. But a common garden experiment where you can just show so much diversity in color and form, all of the same species. And that's what we do in agriculture. So we have gene banks, and arguably, I took it rather literally, the title of this workshop, but arguably, Noah's Ark was the first gene bank. And if you look at where I work, there's some architectural similarities.
Very nice.
But there are about 1,700 gene banks around the world. The USDA's is a large one. There are about 6 million accessions so far. And we house 600,000 unique ones. So it is a very significant collection that we steward. And it's a heavy responsibility, I feel. One of the things that I find really important when we make these collections is that it's not just about having a physical sample. It's about using it. And one of it is people will not request the sample unless there's associated data with it. So we work very hard to make sure that there's phenotypic data associated with it now. With genotypic data, all of that data is available, and there's the site, I think Paul mentioned, GRIN Global, and GRIN started with our database, and now it's expanded to be international, and most of the agricultural gene banks are on a GRIN Global platform. If it's for research, you can ask for it and you will get it. The USDA distributes 250,000 accessions per year. And I think that that is really important in terms of this context. It's not about making collections, and it's not about storing collections. It's about putting them out there, and I don't think you would say that anything about this being a restoration, but it is about understanding what biodiversity is out there and how it can help us understand it, understand the consequences if you lose it. So one of the papers that was very influential to me as I was trying to wrestle with what I do for agriculture and how I can resolve this in a conservation aspect was Michael Soule's paper on conservation targets. And he made it really in a societal construct and gene banking on two axes. One is political stability, where should we put these things, and the other is technological inputs. And a lot of what we've been talking about so far are these restoration projects and where you put them and what are the likelihoods of success on that. And we would call those in situ. And growing them out, trying to get more germplasm, I would put that as an inter situ. We find the sources of material, as we've heard all day today, are difficult. Botanical and zoo gardens, you can see that they're increasing. Usually you see them where there's political stability, but there's also an increasing level of sophistication in maintaining these things out of their natural environment. And there up in the corner is where I live. It takes a lot of This is not a natural environment. So that's my segue into what we do. At our place, we have two different, I would call storage platforms. One is conventional storage, and then the other is cryogenic. The world is very nice to the United States and calls it minus 18 degrees centigrade. That's not because we keep it at minus 18, it's because we don't work in centigrade, we work in Fahrenheit. Minus 18 is 0 degrees Fahrenheit, and that makes it much easier for the engineers to adjust the dials. Liquid nitrogen, well, we use a lot of liquid nitrogen. We have about 100,000 liter habit a year. And this is what I consider germplasm. There's all different forms. Seeds are the most convenient because they're just so easy, lovely to work with. I don't want to call what I do as easy. But we can use anything that a plant uses to regenerate another plant. I've put sperm in there mostly as a place marker because we do have an animal collection. It's all livestock. But pollen would be the counterpart for sperm, and that's where most of the sperm is where mostly is stored in their animal collection, and that would be cryogenic. Plants are so amenable, we can use we can make choices. Okay, the point is to revive germplasm after it's stored. There's no sense for us to be storing dead stuff. So I'm just going to run through some of the basic principles of preservation, just because it's not very familiar. Most people look at alive collections, and I consider our collections quiescent. My background is a little bit of the physical chemistry, so I'll be putting two graphs. One is water content. One of the reasons why our gene bank is in Colorado is because it's so dry, yay for dry, but we're also manipulating temperature. And so the context for most of biology is really, really hydrated. And what we want to do when we preserve things is to put them in the solid state. You all have an intrinsic knowledge of solid state. It's just making something that's fluid and making it hard and have structure. And that is what preservation is all about. So here is, everyone built up my talk about recalcitrance, and we have lots of seeds and a diversity of seed behavior. Everyone thinks seeds are like rocks, but they're not. They're living creatures. But the most of the seeds in the world are what we would call orthodox. They can naturally form these solid states, and that's what they do when they dry down. But a recalcitrant seed doesn't, and it's called recalcitrant just like a difficult child. It's hard to gene bank. So there's also other intermediate. And to understand what these kinds of seeds are, I'm going to take you to Hawaii. John Clark mentioned Hawaii as the endangered species capital of the world. It has more endangered species per square hectare than any other place of the world. So it was quite a feat to understand these and to get these species so that we could actually gene bank them. So the classic orthodox, those are the ones that can form that solid system without a lot of manipulation. Only a third of the species are. That's important because that's our classic way of preserving them. But the recalcitrant, not so bad, only 3%. That means this flora is actually very manageable for gene banking. Then we see a number of other physiologies that we would typically consider as intermediate, and those we're all going to store cryogenically. And I think for this audience, the idea that it was just too difficult to get enough species to do the testing, that was between a fifth and a quarter of the endangered plants in Hawaii. So here's our idea of what we want to do is lower the temperature, In order to preserve this material, what I talked about earlier was just changing the moisture. If we did it with recalcitrant seeds and we just cooled them down, they would form ice and we would kill them. So the idea there is to just play with the moisture contents and the temperatures and get them into a preserved state. And we're pretty good at it. We've been pretty successful with most of the material from temperate areas, so that would be your oaks and others. The tropical material is really difficult. So what I want to just briefly touch on is, what do you buy when you've preserved something? We're talking about buying time, and how much time. Noah had about, 40 days, 150 days to get off. What is it for us? So things age. Even though you've got a preserved state, you have an intuitive knowledge. I pulled this from the library archives, that things do change with time, even though they have structure. We can expect it. It's just what that time scale is. When we talk about germplasm, it's particularly difficult because there's an initial stage where you don't see the change, and then you see a very rapid loss of function. And so we want to regenerate this material and get it out of the gene bank. As I said, what's the use of storing dead stuff? So we want to know what that longevity is, so that that's actually the time frame that you have to collect it and then either regenerate it or use it. So this will be the last segment, and this, I think, I use this when I give tours because it's a very charismatic experiment. You can see from the date on the Life article, it's an experiment that was written up in 1948. So we're getting around 70 years now on this experiment, but it's by a famous plant physiology, Fritz Wendt. And he just asked this question of, how long do seeds live? It's very captivating for someone like me to have it. And I have the honor of curating this experiment. So the samples are in my lab. And it looks beautiful because you can see the paper within the little antique tubes, and the paper is beginning to brown. So we can actually ask that question of what is actually happening to the seeds. So in 2014, we germinated them, all of the samples, and we maintained them. I might be changing a little bit of the concepts of the experiment, but I gave this data for Peter because Peter has a certain affinity for Clarkia, and it's amazing. I mean, we added water, and these things germinated 100% after 2 days, and they were stored at room temperature. So for me, it's a proof of concept that it works. We can do it. You can buy time. And we do have the technology to make this possible. It's a tool. That's all it is. I wouldn't call it a conservation, but it is a tool, and it's a tool available, and I hope you use it. So thank you.